So today I'm going to discuss about what are vaccines. So before going to enter into the topic vaccine, I want to say something about antigens because these are very interrelated topics. You should have a basic idea about antigen. So antigens are such kind of proteins. Mostly these are proteins. It may be simple protein, it may be complex protein in the form of glycoprotein, lipoprotein, or nucleoprotein. So the protein that are complex in nature, mostly having molecular weight 10 kd to 600 kd, can be considered as an antigen if they possess a particular epitope, which is T cell receptor epitope. So these antigens are actually administering either solely or else with a other type of element. Those are called as adjuvants for the production of vaccine. So if I going to define a vaccine, then I must say that vaccines are antigenic material, and this antigenic material can be the whole pathogen or whole microorganism, pathogenic microorganism. It may be virus, it may be bacteria, it may be fungi, it may be other parasite. So if we are using the whole microorganism, then the microorganisms should be live but weakened forms, or else we can use killed or inactivated forms of those pathogens, or else we can use the purified forms of proteins that is actually responsible for the antigenicity of those particular pathogens. So this antigenic material, what is it is doing in our body? So it is actually stimulating the adaptive immune system of our body against a particular disease. So if you are injecting influenza virus protein, surface protein, means you are developing adaptive immunity, you are developing acquired immunity in your system against that particular virus, means influenza virus. So a vaccine is actually stimulating the body's immune system. And how it is stimulating the body's immune system? So it helps to recognize the agent as a foreign particle, then destroy it, and also remember it. So it is also helping the pathogen to remember it. So now the question will come, what is the necessity of vaccination? Why we have the requirement of vaccination? So the requirement of vaccination is that, as because we all know that we always say that prevention is better than cure. So vaccine is actually functioning as an important tool in as preventive medicine, as well as in the public health. So if you want to stop the spread of a contagious disease like swine flu, like coronavirus infection, the pandemic coronavirus infection or swine flu. So vaccination to the mass people is the only way. This is the need or necessity for vaccination. Now, what are the types of vaccines? Vaccines can be classified in different generations: first generation, second generation, third generation. So, first generation vaccines is the vaccine type that Dr. Edward Jenner has started with cowpox lesions to combat against the smallpox. And the same techniques has been carried on for so many years because of the similarity between cowpox and smallpox. And he has used cowpox lesions from the milkmaid and injected the detoxified or weakened live virus form of cowpox to the individuals, and astonishingly found that they are not growing, they are not experiencing smallpox lesions, they are not getting smallpox infection. Then polio vaccine, means live attenuated form of polio vaccine, was also developed. The second form is a killed form. Mostly the virulent pathogen, the microorganism, is killed or destroyed by physical or chemical means. In physical means, you can use drying, you can use irradiation, you can use some chemicals or antibiotics to destroy the virulence part or virulent form of the microorganism, and then you can use it. And the examples are influenza vaccine, cholera vaccine, and these were the earliest vaccines. Means live attenuated vaccine, killed vaccine, these were the earliest vaccines. Then in first generation vaccines I'm talking about, so then the development was on attenuated vaccine. So attenuated vaccine means actually what we are doing here, we are just modifying the microorganism and changing its form or changing its effect. What sorts of effect? So we are reducing the virulence effect or the force of the bacteria or the virus. So Yersinia pestis is one of the example. Then toxoid. So there we are not using the organism as a whole, we are using the component, mostly the toxic compound that should be inactivated, which cause illness rather than the microorganism itself. Examples: diphtheria toxin, tetanus toxin, toxoid, etc. In second generation vaccine, here you can see that to minimize the risk of the live attenuated vaccine, means if attenuation is not properly done, then the virus or the bacteria can cause infection. So to minimize that risk, here we are using live attenuated vaccine. The form of protein antigens such as tetanus, diphtheria toxoid, or else recombinant component of hepatitis B, surface antigen HBsAg. These two, these proteins, surface proteins, or recombinant protein component, they're able to generate the T helper cell and as well as antibody responses. Now I'm coming to the third generation vaccines. These are the DNA vaccines. So here small circular piece of bacterial DNA that is actually encoding the virulence protein or the antigens. So we are injecting this and then it will develop the protein. So we are not injecting the microorganism as a whole, the pathogen as a whole. We are not injecting the protein. We are only injecting the gene. Now what happens after administration of a vaccine? So a vaccine can be administered intramuscularly or subcutaneously. And when you have administered intramuscularly, it will come into the myocytes or the muscle cells. And then the antigen presenting cells, means the macrophages, dendritic cells, monocytes, they will come into play. And the virus, if it is a virus, so virus will internalize via receptor mediated endocytosis. Then there will be proteolytic degradation, means under the endosome, so within the endosome or the lysosome, in the acidic environment, there will be acid hydrolysis of the protein, viral protein occurs, but these proteins will be cleaved into different polypeptides, and the produced polypeptides are then loaded onto MHC1 molecule. Then you can see that the immature dendritic cell will mature the phagocytous virus vaccine, initiating a series of events. So what is actually happening here? This flow diagram will tell you. So via phagosome, via macropinocytosis, via phagocytosis or receptor-mediated endocytosis, they will come here, antigen presenting cell will recognize it and then will go for processing. So here within the endosome or the lysosome, there will be proteolytic cleavage, and then the polypeptide having a particular epitope will bind with T cell, and in a second way, MHC2 and the dendritic cell will mature, will come from endoplasmic reticulum to Golgi apparatus, and here dendritic cells will carry this MHC2 to this T cell, and now they will complex with the protein as MHC2 protein complex. MHC2 protein complex. Now this MHC2 protein will bind with, and the protein complex will bind with the T cell. Here the T cell at the variable region, the T cell receptors, beta chain of the variable region will come into play. When this beta chain will bind with the antigen, then this antigen will be known as super antigen, and if the T cell means CD4 cell plus T cell, MHC2 and the protein will form a complex, then this is a conventional antigen complex. And now this CD4 plus T lymphocyte, which is the helper T cell, will release cytokines, and now the cytokines will induce the B cell to release antibodies and also memorize the event. So I have already explained that adjuvants are used to enhance or boost up the immunogenicity of an antigen in vaccine production. So adjuvants can be of different type of compound. This may be inorganic compound like aluminum salt or gels, which are called alarm. They can be some herbal compound as oils. They can be Mycobacterium tuberculosis detoxified forms, inactivated forms. And these adjuvants will mix with antigen, and they are actually, when entering into the human cell or human body, they are actually governing inflammasome. What type of inflammasome? This is known as nucleotide binding and oligomerization here. This NLRP3 inflammasome. So these are the nucleotides. They will bind with the leucine-rich repeats. So this inflammasome is a nucleotide binding and oligomerization leucine-rich repeat pyrene domain containing 3 inflammasome. It's a full name. So this inflammasome is inducing IL-1 beta, that is interleukin 1 beta. So this interleukin 1 beta will again induce the T cell or induce local inflammation and recruits the APC antigen presenting cell and enhancing antigen uptake, and also responsible for DC maturation, dendritic cell maturation, and stimulation of the T cell. So it will stimulate T cell activation as well as T cell differentiation. Means CD4 and CD8 type of cells, cytotoxic T cells or helper T cells. Then the T cell will induce the B cell to release antibody. So the question will come into our mind that antigen is also doing the same thing. So why should we add adjuvant? Now this slide will tell you what are the advantages the adjuvants are doing in vaccines. So these two steps are almost similar, but here you can see that if you add adjuvant along with antigen in your vaccine, then this adjuvant is also synthesizing or releasing two different type of cytokines. So here they are releasing cytokines. And these cytokines will mature APC, then it will bind with the CD4 T cell, and then it will induce the innate B cell into plasma cell and memory B cell. Memory B cell will memorize all the things, all the steps, all the patterns. Plasma cell will secrete antibody, will produce antibody. But here, these cytokines, they are promoting more number of plasma cells. That is, more number of plasma cells means more number of antibodies coming into your blood and then storing into the lymph node. So by using adjuvant, we are inducing the immunogenicity of the host of the antigen also. So this is the novelty of using an adjuvant with antigen in the preparation of vaccine. Now, how to prepare a vaccine? Mostly vaccines are prepared in vitro culture. If it is a virus, then we have to go for cell culture. So first, here I have given an example of a virus vaccine production. So first, the host cell to be cultured in liquid media. The media may be DMEM or maybe RPMI. So we are culturing the host cell, then we are inoculating the cells to infect the host cell with certain type of virus. You can culture in embryonated egg also, fertilized egg also. Now, different T75 flask, you can culture it, and then you have to harvest the cell. Then in bulk amount, you have to pull it, and then go for centrifugation or purification. So centrifugation and subsequent washing should be performed for purification purpose. And after purification is over, the purified antigen will be mixed with three different other compounds. One is your adjuvant, second is the stabilizer, third is the preservative. The stabilizer is actually stabilizing the structure and function of that adjuvant as well as the antigen. And the preservative is preserving the antigen for and enhancing the vaccine cell life. So this is how the formulation of vaccine will take place. Now after formulating the vaccine, the solution, you have to fill it in different vial, and also you have to seal the vials at once under aseptic conditions. Then you have to check for any impurities there or not. So impurity check or infection is must, because we are injecting it parenterally or else intramuscularly. So whether any dust particle or whether any microorganisms or anything is present there, so we have to uh, check it very carefully. After checking, we have to go for labeling and then packaging to reach to the market. So this is the whole procedure